Dear students, this is Sanjuman Miss. Here I have prepared a module on chapter 11 for the students of class 10 commerce, and the topic has chosen here, that is structure of DNA. Do you know what is the full form of DNA? Deoxyribonucleic acid. It is a compound molecule found in the nucleus of cell. It codes genetic information and passed to the next generation by cell division, and it also helps the growth and development of the living body by cell division. DNA is a double helical structure, that means double stranded structure present in the nucleus. It is just like a compound molecule, like polymer. What is polymer? Poly means many, meros mean parts. When many monomers combine together, then the substance is produced, that is called polymer. Exactly one of there, then the complex carbohydrate starch is produced. DNA is exactly like made up of many small basic unit of DNA which are called nucleotides. So, what is a nucleotide? It is made up of three components: phosphate, sugar and a nitrogen base. Here the sugar represented by the brown color is a deoxyribose sugar, which make bond with one phosphate group and another nitrogen base. What are the nitrogen bases are? That means what are the basic unit of DNA are? Those are actually different kinds of proteins. According to their coding, they are of four types: adenine, guanine, cytosine and thymine. These four nucleotides are also grouped into two. One is pyrimidine, another one is purine. Thymine and cytosine in pyrimidine group, and adenine and guanine are in purine group. Pyrimidine groups molecules are small molecule, but purine nucleotides are large molecule, and that they are arranged one after another to make a long chain. It can be vertically arranged or horizontally arranged, but in DNA the nucleotides are present one after another. And previously we have learned nucleotides are in two groups. And how do they make pair? Always one from pyrimidine make pair with one nucleotide of purine. DNA are highly coiled structure present in the nucleus, and it is a double stranded structure, that means double helical structure. If we untwined a DNA, it just look like a ladder. Two of the sides are considered of two backbones, and two backbones are connected by red color steps which can be considered like rungs and complete the whole DNA structure. Now, how each of the parts are produced? If we think about the backbones, like that means two sides, these backbones are made up of sugar phosphate structure compound. If the nucleotides are arranged one after another, then they get connected sugar with phosphate one after another nucleotides. You already have learned there are 5 carbon are present in sugar molecule. Those are numbered 1, 2, 3, 4 and 5, and here you see the 5 number carbon is attached with the phosphate, and that phosphate is connected with the third number carbon of the upper nucleotides. By this way, the number 5 carbon is getting attached with number 3 carbon of next nucleotide. By this way, the long chain of the backbone is produced of one side. So, this arrangement of the nucleotides follows some rules which can be described as directionality of nucleotides. If we put these nucleotides on the backbone one after another, then it will complete the structure of the backbone of left side, followed by the rule of directionality. So, directionality can be 5 prime to 3 prime or 3 prime to 5 prime. Here the backbone already has produced, that followed the directionality 5 prime to 3 prime, and this backbone is made up of two of the molecules of nucleotide. What are those? Phosphate and glucose. Let us see what happens on the other side of the DNA. Here the nucleotides just moving downward and facing to the downward, and here the 3 carbon molecule, 3 carbon side getting attached with the nucleotide phosphate group, and that phosphate group is getting attached to the next nucleotide with their 5 carbon. So, the directionality of this nucleotide is 3 prime to 5 prime. By this way, two of the sides of the DNA is produced. Now, come to their base pairing of the nucleotides. Actually, they are pairing with their nitrogen bases, and always here follows the rule, one from pyrimidine and one from pyridine nucleotides will make bonds. So, you already have learned the purines are large molecules. So, always one large nucleotide, one small nucleotide will make pair, and if that means adenine, if adenine make pair with thymine, then cytosine will make pair with guanine. Then keep the balance of the structure from beginning to the end, and you know our nucleotide can contain our DNA can contain 2 to 1600 of base pair, that means nucleotides, to complete its structure, and there are hydrogen bonds present in between of them to get attached. In between adenine and thymine, 2 bond of hydrogen is present, and in between of cytosine and guanine, 3 bond of hydrogen are present.